Good morning, everyone. My name is Dr. Smith, and this morning we shall have a brief news on the latest technological advancement in the medical field, mostly on the areas of gene editing or gene therapy. Of recent, the government announced the enrollment of lenacapavir, which is a drug which can help in preventing the spread of HIV. Lenacapavir is a long-acting drug that has a greater efficiency, mostly for HIV type 1. It has less affinity for HIV type 2. However, majority of people have a misconception about lenacapavir as a vaccine. This is just a long-acting drug, which is a capsid inhibitor. Capsid is the viral outer coat. Normally, when the virus, after replicating within the CD4 cells, that cell wall, the capsid splits and the virus is released into the blood system, which then goes to infect other CD4 cells. Now, lenacapavir has that capability of preventing the capsid from breaking down and releasing the virus. The drug is capable of being effective under low concentration. The rate of metabolism of lenacapavir is relatively low, which makes it very effective to be used over a long period of time, like 6-month interval between. The next discovery in line is in gene therapy is the discovery of sickle cell gene therapy medication. The first one we can talk about is called Gasgivy, or the Exa-cel, which prevents red cell sickling. You know, in sickle cell, normally the main issue is under certain conditions, you find that red blood cells assumes a sickle shape, which prevents its ability to carry oxygen. And this has been the greatest challenge with those with sickle cell. You find that during a sickle cell crisis, when their blood cell sickles could not really carry enough oxygen, leading to what we call asphyxiation or tissue hypoxia. Some of these red blood cells, because they are sickle-in shape, when they reach narrow blood vessels like those within the bones, they cause a lot of pain because the blood vessels will be blocked. Another drug under that line for sickle cell gene therapy is called Lyfgenia. Lyfgenia is also called the love girl. Its mode of action, majorly is that it promotes formation of functional hemoglobin. The functional hemoglobin, the other one is trying to prevent the hemoglobin or the red blood cells from sickling. Meanwhile, the love girl is ensuring that the bone marrows generate red blood cells which are in normal shape, which can easily promote oxygen carriage. The challenge with these medications is that most of them are still in the Western nations, in UK, in America, and they are very highly expensive because a single injection of Lyfgenia is about 2.3, 2.1 million US dollars. An equivalent in our Uganda currency, that would go for around 600 to 700 billion, which is a lot of money. Majority of the average Ugandans may not be in position to have it. The next discovery on the medical field was Zolgensma. It is also a medication used for gene therapy, mainly for the treatment of spinal muscular atrophy. Spinal muscular atrophy is a genetic condition that comes about as a result of a discord or failure of the gene which is responsible for martial response, that martial movement. Normally, when this gene is defective, it could no longer control martial movement, resulting into severe martial atrophy or martial deformity. Your nerves can no longer respond, you cannot walk, results into you being crippled. So this Zolgensma, also a single injection, can help you to get well. Now, gene therapy is a technology which enables doctors in the current practice and medical field to target specific genes within the DNA which encodes for specific proteins responsible for certain physiological activities or response like martial movement. And since this technology has been discovered, the medical field has been able at the moment to correct some junk DNAs. So when you have like this sickle cell, there is a gene that encodes for production of red blood cells which are already sickler. So the gene editing technology enables medical practitioners to get into the DNA and remove that defective gene and replace it with a normal gene, and the body can function well. Next up in line is the attempt to discover a gene therapy for HIV. The challenge for HIV, why it has been very hard to discover a particular vaccine or a drug for it, is because of the high rate of mutation. Now, using conventional antivirals like dolutegravir or the TLD, which is commonly in practice, the challenge is HIV can be cleared by these the replicatory forms or the HIV viruses which are active in the body are normally destroyed by the antivirals. But HIV has a capability of undergoing a dormant period, a dormancy stage, in which they move into the CD4 cells, or mostly the dormant CD4 cells, which are capable of remaining dormant for over 10 years within the system. And when they detect the concentration of the antiretrovirals within the blood has reduced, they release themselves from this dormancy period. Normally, the genes that codes for emergence of dormant ARE virus are the GAG genes and the POO genes, which are able for producing viral specific enzymes that helps in reactivating the enzymes within the system. Now, scientists at the moment are working on a way to edit these genes, remove the GAG and the POO genes from the dormant CD4 cells, and the cure can be elicited. The main challenge at the moment with HIV, you find that if under normal circumstance you are supposed to be cured of HIV, that could take a period of over 80 years, 60 to 80 years, as the virus is undergoing natural death through the dormancy of the CD4 cells. Thank you so much, my viewers. Like and share so that this information can go to a good majority of people. Thank you. See you next time.